Great. Yeah, thank you very much. Thank you for the introduction and thank you for the invitation. I'm really excited to be sharing my work, or really my group's work on this platform. This is certainly something new for me, but very happy to be here and join everyone in the virtual space. So today I'll be talking about some of our group's work on layer by layer coatings of collagen and heparin to control and enhance cell behavior. And before I get started, I do want to highlight really my research group. So they are the ones who do pretty much everything. They're the ones who are doing all the great work that I will be presenting. I have several graduate students whose work I'll be highlighting, including Luis Massa and Hemanta, and also a former grad student who's currently at the University of Texas, Austin, doing a postdoc, David, who's a lot of his work is there. Also, big thanks to all my collaborators at the University of Arkansas and the University of Puerto Rico, at Georgia Tech, and at Integral Life Sciences. If you want to know more about me, feel free to reach out. You can email me. I'm on Twitter as well. Our Twitter is mostly, or my Twitter is mostly to promote our group's work. My website is in this slide as well, so feel free to visit there. I got a description of our projects, our papers, publications, and whatnot. And then really importantly, if you go to my website, you'll see this image, which is really what we believe in what I personally believe and what we believe in our lab. We are a very inclusive lab, open to everyone, and so really we'll just welcome everyone. So if any of you are out there are looking for a research lab, go ahead and contact me. All right. Before I get started on the research, I thought I'd talk a little bit about my journey here. So I'm from Puerto Rico. I was born and raised there. After high school, I went to Iowa State to pursue a bachelor's degree in chemical engineering. Afterwards, I went to Colorado State to pursue a PhD in chemical engineering. And then after, I went to France at the Grenoble Institute of Technology to do a postdoc in mostly biomedical engineering. And then I started my career as a tenure track assistant professor at the University of Puerto Rico, Mayagüez, in chemical engineering as well. And since 2018, I've been at the University of Arkansas in the Department of Chemical Engineering as well. So again, you're hearing chemical engineering over and over and over again, which is true. It is my academic background, but really my research has been more at the interface of chemical engineering and biology and materials, sort of more in the biomedical engineering realm. And so a few examples of our projects that we have going on in our lab are shown in this slide. And I'll be focusing this talk on this two examples here. The first one on enhancing nerve guide conduits to improve regeneration of nerve tissue, and the use of coatings to enhance cell response. However, excuse me, I do want to give a shout out to this other project on microneedle patches for drug delivery, where my student Catherine will be presenting here on the 17th of December on this exciting work that we're doing on developing materials for drug delivery for cows. All right, so to get started, some of the challenges that in healthcare that we are trying to address. One of them is peripheral nerve repair. Why is it important? Well, there's a bunch of surgeries done per year and a huge market as well. And, when I'm talking about peripheral nerve, I'm talking about any nerve in our nervous system that's not the brain or the spinal cord. So you can imagine any injury that you could have to any extremities or any disease that would damage nerve. That's what we're trying to address. The other area of research that we're focusing is on cell therapies. And cell therapies is a fascinating field where essentially you're using cells as a drug, basically, to treat diseases. So you can imagine now that the cells are living entities that we're trying to harness them as a therapeutic, but they're still cells, right? So they are they have you cannot treat them exactly as you would treat a chemical drug during the manufacturing process. So in the U.S., there's a couple of FDA-approved cell therapies. There's many more around the world, and it's a growing market. I mean, it's just incredible. Right now, it stands at about $4 billion, with a projection of about $180 billion per year, which is just amazing. And so, what are the challenges there? Well, one of the challenges in nerve repair is that while there's what we call nerve guide conduits available that would promote the regeneration of nerve, they have their limitations. And particularly when you're talking about large sections of damaged nerve, those conduits fall short. So there's a need to improve their repair capacity. With regards of cell therapies, I sort of hinted on what the issue is that you're trying to now manufacture large scale cells that will ultimately end up in our bodies as a therapeutic. So really, there's not a standard practices. There's not, there's a lot of variability between the different researchers and developers and industry with cells. And a big issue with growing cells at this large scale is that all of the materials that we use are really far removed from their native environment. And there's a lot of research out there that shows that affects the efficiency of the cells, or therapeutic efficiency. And so that's what we're trying to address as well. So how do we actually do that? Well, we use as our inspiration the native cell microenvironment, or what it's known as the extracellular matrix, the ECM. So in our bodies, cells are surrounded by this fascinating fibrous matrix that's composed of all sorts of things, polysaccharides, proteins such as collagen, a bunch of smaller molecules, water, etc. And all of that is organized in a way that it provides both mechanical cues and biochemical cues to the cells. And all of those cues really control all sorts of different cellular processes, such as cell adhesion, cell proliferation, cell migration, and differentiation. And what's important about controlling those processes is that ultimately that leads to tissue formation and tissue regeneration, or even disease treatment, right? And so our idea is that, well, can we capture some aspects of this extracellular matrix and provide some of those cues to cells to be able to control their cellular processes. And so our approach in our lab to do that is well, we have several techniques. The main one that I'll be talking about today is layer by layer. So layer by layer is a very simple yet powerful and versatile technique to do surface coatings. It's illustrated here. Essentially, you have the surface that you want to modify, that it has an electric charge. You expose it to a solution, a polymeric solution containing a oppositely charged polyelectrolyte. Due to the electrostatic interactions, the polymer gets deposited. We rinse it to remove any loosely bound polymers, and then we expose it to another solution of the oppositely charged polymer, which again, it gets deposited on top of the previous one. You keep doing that over and over again to however many layers you desire. So the cells, or what we do then, is that we coat surfaces on this using this technique, and then we culture cells on them, and we see how the cells respond. In particular, we study how the cells respond to different cytokines and different growth factors. And what I'll show you today is some interesting results on that behavior. And here you can see some of our publications where a lot of the work I'll be showing today is available in more details in those publications. A few other things that we're doing in our lab, as I mentioned before, is working with microneedle patches. That's mostly for drug delivery, but we also work with electrospun polymeric nanofibers. Though I won't be talking a whole lot today, we've had some extensive work on developing this type of nanofibers as biomaterials for bone regeneration and as models for cancer cells. But we also actually have a very good collaboration with a colleague here at the University of Arkansas on developing fibers for water treatment as well. All right, so like I said, I'll be focusing this talk on layer by layer films. And so the key players in our films is in our layer by layer films are these two polymers, so collagen and heparin. So why do we use them? Well, collagen, I think most of us know about collagen, right? So it's a protein. It's the most abundant protein in our bodies. It provides structure to a bunch of tissues. And what's more, perhaps to me, what's fascinating about collagen is that at the conditions that we use it and the pH that we use it at, it behaves as a polyelectrolyte. So it has an overall positive charge. The other polymer that we use is heparin. So heparin is a glycosaminoglycan, sulfated glycosaminoglycan. It's an important component of the extracellular matrix as well of tissues. You may have heard of heparin as an anticoagulant, which is by itself a nice property, but really that's not what we use it for. Instead, we use heparin because of its ability to bind many growth factors. And there's a lot of work out there that shows that growth factors that are bounded to heparin, they're protected from degradation, and they can even their activity can even be enhanced when heparin is present. Heparin is a negatively charged polymer, polyelectrolyte. And so that's it. That's why that's our polyelectrolyte pair combination that we use. And of course, both of them being natural polymers, they're biodegradable, right? So when our bodies and when they degrade, their products are pretty much inert. so they won't cause any sort of immune reaction. All right, so I'll talk a little bit about layer by layer films. So again, how do we do them? We work a lot in cell culture with multi-wall plates that they look as shown here. So the way we construct the multi-layers or the layer by layer films on these plates is that we add to the well one of the polymers, say heparin. We let it absorb for a couple of minutes. We remove it. We add a rinse without any polymer. Rinse it a few times, remove it, and then we add the collagen and let it wait a couple of minutes for the collagen to absorb. And then we remove it, then we rinse, and then we repeat over and over again. And as we do that process, the polyelectrolytes gets deposited on our surfaces due to the electrostatic interactions. And I'll only show this slide for characterization, but we have done a very extensive chemical and physical characterization of these coatings, and we've demonstrated that they're quite stable with temperature. The coatings are in the nanometer range. So with the number, obviously depending on the number of layers that we use, but with the number of layers that we typically use, the coatings are about 70 nanometers or so. But yeah, to confirm that we can actually build these films, I'll show you here two sets of data. So first is a set of potential data that shows us how we can invert the surface charge by simply changing the terminal layer. So as we keep adding layers from heparin to collagen, we go from negative to positive, and so on and so forth. We also use a colorimetric dye known as Taylor blue, which binds to heparin, and that's what is shown here. And as you can see here is that as we deposit more bilayers, more heparin gets deposited, and we can detect stronger absorbance. That gives us an indication that that the multilayers are being formed. And if you can see, this absorbance is fairly linear. And we've done studies with ellipsometry that indeed it shows that the growth of these films are pretty much linear in the nanometer scale. All right, so on to the first project. The coatings that we use are, or the idea here is to develop this heparin collagen coatings to be used on nerve guide conduits. So ultimately what we want to do is to have these nerve guide conduits and be able to coat them and show that the coatings improve the behavior of the conduits. Again, the conduits is essentially this tube, basically, that you would connect the two ends of the nerve. So the idea is that, during normal nerve regeneration, if you have a damaged nerve, first, cells go ahead and create basal lamina, and they start secreting a bunch of neurotrophic factors, and that promotes axonal growth and to connect to the damaged stump. So if we can replicate that by having a scaffold, say the nerve guide conduit, that in conjunction with the neurotrophic factors and using our layer by layer coatings to serve sort of as a basal lamina, then perhaps we can improve that regeneration and ultimately have a new strategy for nerve repair. And so that's what we seek out to do. And with what I'll be showing you in the next couple of slides is a bunch of in vitro work with different types of nerve or of cells from the nerve system, showing that indeed our coatings they work. So what are we going to be seeing here in the next couple of slides? So we're going to be investigating the use of nerve growth factor, NGF, which has been shown to be really important in nerve regeneration. So NGF, like many growth factors, has a very short plasma half-life. And it is known to modulate neurite extensions. And so there's Schwann Cells type PC12 cells that we'll be using to show that. And they've also been shown to modulate the behavior of Schwann cells. Now, Schwann cells are a very important type of cells in nerve repair, and I'll be focusing a lot on them. And they are the ones in charge, are partly in charge of the healing process of damaged nerve. So they provide new myelin sheath. They secrete a bunch of molecules to recruit cells to help with the regeneration. And then after they regenerate the tissue, Schwann cells are in charge of maintaining neurons healthy. Okay, so they're really important cells in our nervous system. All right, so some of the results that I'll show you that I'll have are shown here. So with the PC12 cells, so again, these cells, they respond to NGF by doing neurite extensions. So if we look at our control surface, when we don't have NGF, we see the cells that they're attached, but they don't do any neurite extensions. But in the presence of NGF in the control, we start to see some short neurites. Well, when we do the same on our coatings, first of all, we see that in our coatings, the cells do attach. So that's pretty good. But then when we add NGF, whether we add it in solution or if we incorporate it as part of the coatings, we see really long neurite extensions. And that's quite favorable. So that tells us that the cells really do like these surfaces, and that the NGF is perhaps acting in a better way than our control surface. All right, then we went ahead and investigate the behavior of Schwann cells on these surfaces, and we performed initially a viability study where essentially we culture the cells on our control surfaces, polystyrene with and without or with NGF, or in our coatings with and without NGF. And interestingly, we saw that, first of all, there wasn't a huge difference between the conditions of with and without NGF in either surfaces. That's okay. But we see a very drastic difference in viability in the conditions where we have our coatings, our heparin collagen coatings, compared to the uncoated polystyrene control. And that was really intriguing to us. And so we decided to investigate further. We did some proliferation assays and whatnot, and saw that it wasn't necessarily due to cell proliferation. And so we decided to do this study using this apparatus that basically is an impedance-based system where the cells are cultured on an electrode, and the impedance is recorded as a parameter known as the cell index. And so as the impedance or the cell index increases, that means that there's more cells in contact with the electrode. And the powerful thing about this apparatus is that we can monitor the behavior of the cells in real time. And so what we saw, it was quite interesting. So in our control surface here, we see that, the cells attach and they grow, and they seem happy. And then when we add our coatings, though, we see a very significant improvement in cell attachment. And that led to a significant amount of cells growing and reaching that plateau of growth much earlier. But then when we added the NGF, we saw a huge spike in cell attachment to the point where so many cells attached that they had to deattach as well because there was only enough surface cover. And then eventually it reaches plateau as well. So really what this data tells us is that our coatings, perhaps what they're doing is that they're helping, they're improving that initial cell attachment. And in a sense, they're also improving that response to the NGF, which the NGF may, along with the coatings, also significantly improve cell attachment. And that, of course, more cells that are attached, then that would lead to higher cells at the end of the day. At the end of the day, and in the context of nerve regeneration, I mean, that's important because, again, these cells, these Schwann cells, are really important for nerve repair. So the more that you get in that area, well, the better. All right, so we did another study to sort of see what was happening, and in this study, we did a migration assay where essentially my student 3D printed this barrier where this tip here would go at the bottom of the well. And so you would seed cells all around that tip, and the cells would attach and grow, but they would not enter that area where the tip is in contact with the well until you remove that. And once you remove that, then we can observe over time how the cells invade that empty area. And what we saw is that in the control surfaces, after 70 hours, we have cells that go there, but they don't completely cover the entire surface, versus our coatings, we see a complete coverage of the empty area. And so that was quite fascinating to us. So not only the surfaces are improving cell attachment, but they're also improving cell migration as well. Again, another very important parameter in nerve regeneration. If you have cells that are migrating quicker, they'll be able to heal that tissue in a faster way. We did some protein expression analysis, and I'll show just two of them, IL-10 and BDNF. IL-10 is responsible to the anti-inflammatory response, so an important protein to have during tissue repair. But perhaps more importantly is this BDNF that is known to promote the extent of myelination in our tissues, in nerve tissue. And what we saw very interestingly is that the expression of BDNF increases drastically when our coatings are present. We saw that both at 3 days and even more so after 6 days of culture. Drastic increase of that protein expression, and that's that's really good. That tells us that the cells in our surfaces are in a state of repair, that they want to repair, they want to secrete all of these factors and promote nerve regeneration. All right, so to wrap up in this project, we wanted to see if we could do this coatings on an actual nerve guide conduit. And for that, we use Integra's Life Science NeuroWrap that is shown here. And I'm just going to show you just some quick SEM images before and after doing the coatings, just to show that the morphology really of the scaffold is not changed. Again, keep in mind that these are nanometer thin coatings, so we don't expect the coatings to affect the bulk properties of the scaffold. But then when we culture Schwann cells on the scaffold with and without the coatings, we see some very interesting results. Again, measuring just the viability. In the blue here are conditions with our coatings, and we can see that cells are more viable with the coatings, regardless if we have NGF or not. So again, very favorable result as well. And this is a project that we're continuing to explore different avenues with this project, but overall we're seeing that this coatings really do improve that cell behavior, that Schwann cell behavior. So yeah, so to wrap up this project, again, hopefully I've been able to show you that we can not only create this type of coatings, that we can make them on this nerve guide conduits, but more importantly, that these coatings, they can really modulate in a positive way the behavior of Schwann cells and the response to the nerve growth factor. All right, so moving on to the next project. So for this next project, we're going to be focusing now on a different type of cells, and these are MSCs, or mesenchymal stromal cells. So MSCs are a fascinating type of cell that they secrete a bunch of cytokines, chemokines, and growth factors. They also secrete niche molecules such as MMPs. And you can differentiate these cells using specific type of chemistries and proteins and whatnot. And all of that is important because all of the secretion of these cytokines and chemokines, they can be used to reduce inflammation in cell therapies. They can also be used to modulate immune cells or do some immunomodulation. And they can be used to promote angiogenesis or the formation of blood vessels. The niche molecules also, or the MSCs through the niche molecules also promote the regeneration of tissues in our bodies. And perhaps what are they most commonly known for is that these cells, they can differentiate into bone, into fat, into cartilage, into many other tissues as well. And from a manufacturing standpoint, one nice thing about these cells is that they self-renew. We can expand them in culture. So MSCs are quite an attractive cell for cell therapies. There's a bunch of studies out there investigating the use of these cells to treat a number of diseases. And at the same time, there's a lot of studies out there on how can we expand these cells in a larger scale. And so in a typical manufacturing process for the cells, so normally you collect tissue from the patients that the cells are located. Usually it's either bone marrow or adipose tissue. You process that tissue to separate the cells, and then you expand those cells and create a cell bank that you freeze. That initial cell bank, then you can thaw it and further process it to expand each vial into many more vials of product that can ultimately lead to back to the patients. Now, all of these processing steps have been shown to reduce the potency of these cells. And one theory behind it is that all of these steps are done in typical bioreactors that are hard synthetic plastics that are really not what the cells are used to in our bodies. So for example, some the different types of bioreactors that are out there. You can use planar and multi-layer stack systems, again, hard polystyrene surfaces. You can use whole fiber reactor, or you can use stirred suspension-based bioreactor where the cells are cultured on spherical microcarriers that are suspended by an impeller. So our idea then is, can we use our coatings to coat this different type of bioreactors, and would that improve the efficiency of these cells? And so that's what we set out to do. So, we coated cell culture plates using our layer by layer coating. Same approach using collagen and heparin. And then we seeded cells on them, and then we study the behavior of these cells with another important cytokine that I'll talk here in a second. And this images that I am showing here is just to show you the cells, the MSCs, on our control polystyrene surfaces and on our multi-layers or layer by layer films that are either terminated on collagen and heparin. And this just shows you that morphology-wise, the cells, they look pretty good. They pretty much look just as good as our control. All right, so we wanted to study the effect of this cytokine interferon gamma. And interferon gamma is a fascinating cytokine that it activates the immunosuppressive properties of MSCs. And one way that it does that is by activating the production of this molecule known as IDO. It's been also shown to enhance the regeneration of tissues. And what's nice about IFN is that heparin has been known to protect this cytokine from degradation. It provides stability. So we did cultures for the cells on our coatings in the presence and absence of this cytokine. And then we did a bunch of studies. So again, first starting off with just looking at cell morphology. Basically, we see no difference in the presence or absence of the growth of the cytokine of IFN. Cell morphologies, they look pretty good. Then we investigated proliferation, and here's where we start to see some interesting behavior. This protein, this IFN, when we expose the cells to the IFN on our control surface on polystyrene, we actually see a decrease in proliferation. And so that's not ideal, right? So if you're manufacturing the cells and you want to treat them with these proteins to enhance their therapeutic property, you still want them to grow, right? And so you don't want their proliferation to decrease. So we did the same with our coatings. And what we observed, so we saw a slight decrease again in proliferation with our collagen terminated coatings, but on our heparin terminated coatings, we see no difference. We see that the cells proliferate just as well in the presence or absence of the interferon gamma. And so that was quite positive to us, because what we think then is that perhaps the coatings are interacting with the protein in such a way that it negates this anti-proliferative effect while maintaining the immunosuppressive properties. So we did some more studies. We did an analysis, immunofluorescence of the focal adhesion kinase. So this is an important receptor in the cells that is involved in cell adhesion. And at the beginning, we saw that really there's no difference in the expression of this kinase in the presence or absence of the growth factor or in the different surfaces. So in the images here, the expression of the kinase is the green dots, whereas the blue is the cell nucleus and the red is the cell cytoskeleton. All right, so then we went ahead and did another study with that impedance-based assay. And again, here, similar to the Schwann cells, what we saw is that, you know what, there is a difference in cell adhesion. The curves here in blue and green are conditions where we have our surfaces, our collagen heparin coatings, and they are the cells adhere much much more strongly and than in the uncoated surface. And we saw this with cells from two different donors. So our surfaces really do something to that cell adhesion as well. We investigated protein expression and this heat map with these 4 different proteins that are very important in the immune properties and also in tissue regeneration, in angiogenesis, for example, VEGF. And we saw again that our surfaces have a very, they promote the expression of all of these proteins, particularly heparin-terminated surfaces, and even more so in the presence of interferon gamma. So heparin-terminated surfaces, as shown in the top 2 rows, and particularly with interferon gamma, have a really, really high expression of this, of all of these proteins. So that was quite favorable. That, you know, tells us that, you know, in fact, the cells are behaving differently with these surfaces, and perhaps in a positive way. Then we were interested to see the immunosuppressive property. And the property that we wanted to measure is IDO. And so what happens is that when the MSCs are exposed to the interferon gamma, they are activated into anti-inflammatory MSCs, and they start secreting this enzyme known as IDO. And that enzyme is a known immunomodulator. Okay, so it affects that enzyme affects the behavior of cells in our immune system, and that can be a good thing, especially if we're using MSCs as treatment, because that means that our immune system won't go and quickly attack these cells. Rather, they'll let them be and have these cells do their job. And so what we see is that the expression of IDO, again, where it's higher when the cells are exposed to interferon gamma. That's expected, but it's significantly higher when the cells are in our surfaces. And this was true for 2 out of the 3 donor cells that we investigated. So that was quite favorable as well. Again, heparin-terminated surfaces being sort of our best surface that promoted a large secretion of this molecule. We wanted to make sure that our surfaces don't affect the differentiation of these cells, and so we went ahead and differentiate them toward the estrogenic and adipogenic lineages. And we use different stainings, alizarin red and oil red O. And basically all of these images show you that the cells have the capacity to differentiate. And we did another assay, alkaline phosphatase assay, showing that, again, the cells basically can go towards the osteogenic lineage. So, you know, another important function of the MSCs is their capacity to differentiate, and lo and behold, they can still do that. Lastly, we wanted to compare, okay, you know, is it really something about the layer by layer films, or is it just one component versus the other? And so we did a study where we did cast films of just collagen or just heparin, and we study cell viability, both using adipose-derived MSCs or bone marrow-derived MSCs. And what we saw, in fact, was quite interesting is that when we used the surfaces alone, either collagen or heparin, not together in a layer-by-layer fashion, the viability actually decreases. And that was quite interesting. So there's something about that layer-by-layer interaction that the cells rather like, as opposed to just doing the just one coating of collagen or one coating of heparin. So what do we think is happening? Couple of things. The layers themselves might be acting as a reservoir for the cytokines, for the IFN, basically allowing the IFN to absorb on the surface and getting concentrated. And as more and more IFN absorbs, it activates more and more receptor. That certainly is one theory behind it. Another theory is that the IFN is actually interacting with the heparin, as shown here, and that interaction actually allows for the activation of different types of receptors that translate into a different cellular response. And so we have some NSF funding to explore those possible mechanisms, and hopefully we'll be able to sort of discern what's happening here in the near future. And then sort of my last results slide. So I'm, started talking about how we want to use these coatings for manufacturing. And so indeed, really that's what we want to do. And so the platform that we're mostly interested is this, if you recall in my previous slides, this idea of suspension-based stir tank with microcarriers. So the microcarriers are these spherical beads that the cells grow on these beads, and because of their high surface area, you can have a lot of cells grown on them. And so what we do, we've developed different coating techniques, different layer-by-layer coating techniques of these microbeads. One of them, for example, is this centrifuge-based assay where we suspend the beads on the polyelectrolyte solution, let it absorb for some time, centrifuge it, remove it, rinse it, centrifuge, remove, add the collagen, centrifuge, remove, and then rinse it again, and you keep doing that over and over again. And that actually gives us some really nice coatings. So here in the green here is actually fluorescent collagen, and we saw a really nice uniform coatings on these polystyrene beads that are shown in blue. And the cells attach, and they like these coatings. So we're expanding this work more and more, and moving into larger scale cultures. So to wrap up, hopefully I've been able to show you that these coatings can be used for cell manufacturing, particularly MSCs. All sorts of different properties of the MSCs, adhesion, proliferation, protein expression, and response to IFN are enhanced in the presence of this coatings, and particularly that the IFM seem to have a particular preference to heparin-terminated coatings. With that, I'll wrap up this presentation, going back to this picture, and hoping that you can take away that we've been able to sort of mimic at least part of the extracellular matrix, at the very least at the chemistry that the cells are sensing in a 2D level, by using this layer-by-layer coatings. And we see that in fact these coatings, in spite that they are nanometer thin coatings, they can actually modulate cell behavior. With that, I want to acknowledge again my research group, my collaborators, and the different funding sources. including the Arkansas Bioscience Institute, National Science Foundation, the Oak Ridge National Labs, Oak Ridge Associated University, and the USDA for the microneedle patch work that I touch upon. Again, really a big shout out to my students. I mean, they are really the ones that did all this hard work. Here, there's some more of my grad students and undergrads that have work on the stuff that I showed today. And lastly, I just want to thank you all for listening to me ramble for a couple minutes here, and happy to answer any questions.
Wonderful. Thank you for a really interesting presentation. So we'll just go ahead and jump straight into the questions.
Sure.
As a reminder, anybody that, there's not a cutoff on the questions, feel free to keep asking them as we go. So the first one here, how large a window do we have to try and repair these damaged nerve endings?
Yeah, so it's in the order of centimeters. So I can't recall the exact value, but if it's a centimeter or less, this type of scaffold work fairly well. Is it more? Then you start seeing failure, and basically failure what it means is that it just never breaches, right? So
okay.
So you may need to do either a second surgery or add some more drugs or whatnot to sort of improve the process. So we're trying to avoid that by simply using our materials to improve that regeneration.
Gotcha. Okay. In terms of the coatings or the coatings that you get from the layer by layer process, how complete are those coatings, and how many layers are required to get to what you might consider 100% or as close as reasonably possible coverage with the layer by layer process?
Yeah, no, that's a great question. So one of our papers actually explore that. Now I'll say that this depends strongly on the underlying surface, both on the charge and on its morphology, topography, and whatnot. But nonetheless, we did study with, say, both glass and silicon wafers. And if I recall correctly, I think it was pretty much after 4 bilayers.
Okay.
4 to 5 bilayers, we have almost complete coverage. So our standard is either 6 bilayers or 6.5 bilayers. So basically 12 layers or 13 layers.
Yeah.
And that pretty much gives us complete coverage.
Okay.
I was going to say, and keep in mind that when we did the study, we measure the coverage on dry films, and we have another paper that's coming out, hopefully in the near future. That we did some, and this was done using atomic force microscopy, using some wet films, some atomic force microscopy under solution, showing that it's pretty much the same. I mean, the films swell, sort of like a hydrogel a little bit.
Okay.
It's like a nanogel.
Yeah.
But still, they have pretty good coverage after those 6 bilayers or so.
Okay, nice. So I'm actually going to skip to another question that came in a little bit later, but I think is related. Is there a point of diminishing returns in the number of layers? So if 13 is sort of the number that you need to get the full coverage. If you kept going, does it start to, I guess, have a negative effect?
Not really. I think. Okay. So and this is mostly so I've been doing layer by layer work for pretty much all of my career. So with this particular system, we haven't explored a large amount of layers. But with other systems that I've worked with in the past, what happens is, say, when you start getting into the hundreds number of layer, now you go from a nanometer thin coating that you can hardly see into a micrometer, even millimeter sort of scaffold. Okay. You can get what's known in the layer by layer film as a freestanding film, something that you can actually manipulate by hand and use it
Mm-hmm.
as a scaffold. So
Okay.
so basically, what this technique allows you is to either do surface coatings, or if you keep going and going and going, it allows you to actually create scaffolds. And obviously, at that point, you're talking about properties of a material at the nano scale versus the micrometer millimeter scale.
Yeah.
And the mechanical properties are all sorts of different at that point.
Sure,
right?
that makes a lot of sense. Let's see. So I think this was referring to the images that you showed with the growths from the cells. How long does that growth take?
Well, I guess it depends on which ones exactly. But typically our studies are done for either, let's see, for the nerve cells, we do them 3 to 6 days. We've done up to a week or so. For the MSCs, we've been doing a little bit longer studies now that we're focusing more on this larger scale manufacturing processes.
Okay.
I think my student has been able to keep them for 2 weeks or so.
Wow.
At the larger scale. Now, of course, when we're doing the cell differentiation, so that differentiation process takes quite a long time.
Okay.
And so there we also do at least 2 or 3 weeks of culture. So yeah.
Okay. Let's see. Okay. Could you alter your coatings to enhance cell adhesion further, so that the spike results in a higher steady state cell index?
Yeah, I mean, possibly. So one thing that we could potentially do is just add more adhesive moieties, right? So there's a bunch of proteins that promote cell adhesion, such as fibronectin, that could certainly be added there, or even peptides. So there's a peptide sequence, RGD, that's known for cell adhesion. But keep in mind that peptide sequence is actually in collagen, right? So really, collagen is the key player here, modulating the cell adhesion, we believe. So, I mean, how much more we can enhance it, I don't think a whole lot more, but maybe. Yeah.
Okay. Is there an advantage to doing that? Like, is having more cell adhesion a good thing?
I mean, in my opinion, it could be, right,
Okay.
in the context of tissue repair, right? So, if you imagine that you have more cells that are attached during that initial repair process, then those are more cells that can then divide and then, repair.
Sure. Okay. Yeah,
So
that makes sense.
yeah, but that's my opinion, at least.
Okay. Let's see. On slide 21, if the cells are viable without NGF, does that mean you could do something with them without NGF, or is it still needed to drive the growth?
Yeah. No, yeah. So let me just go back here if I can. I mean, short answer, yes. Initially, we wanted to sort of we wanted to use NGF, right? But then we were really happy to see that we may not need it, right? And that's even better.
Yeah.
So, and of course, we can explore other factors and drugs that are related to nerve repair, and that's something that we want to do in the future.
Okay.
But the fact that just the coatings alone did so well, that was really, really encouraging to us.
Makes a lot of sense. Yeah. Okay, this is a question I had, because I do a little bit of impedance spectroscopy,
Sure.
actually for other work, but it kind of with a similar purpose.
Sure.
Does the cell index number correlate linearly with cell adhesion, or is it linear for part of it and then it kind of rounds off?
So I guess I didn't fully went into it, but it's not only just cell adhesions, right? So the different, so the way we need to interpret this data is really based on knowledge on how the cells behave over time. Okay. So we know in the first 10 hours, maybe up to 12, 18 hours, we know that they're primarily undergoing adhesion. And so we can say that whatever behavior we see here is related to cell adhesion. Okay. Now, once that they adhere, say after 20 hours or so, then the cells start growing and proliferating. And so this growth that we see here is no longer due to cell adhesion, but actually due to cells that are duplicating and growing, right? So
got you. Okay.
So that's sort of, that's and yeah, I didn't expand this a whole lot in the discussion, but it's important to have an understanding on how the cells behave, because not all the cells adhere in the same time window, right? Some of them are much quicker. Some of them take 24, maybe 48 hours. So
okay. That's it. So, yeah. Okay, that's actually really cool. That makes a lot of sense.
Mm-hmm. And that's the nice thing about this equipment, is that it allows, because we can keep it running for forever if we wanted to.
Yeah.
At a point the cells are eventually going to die, but it allows us to really monitor through a really long period of time what the cells are doing, and knowing sort of how they behave, then we can sort of correlate those different changes in cell index with the different processes. Yeah. And so, for example, if at any given time we were to add something that's actually, we do this quite a lot, if we were to add, say, a drug or a material, and we want to see how the cells respond, then we can see, if we see a decrease, then the cells are dying. If we continue to see a further increase, or even a sharper increase, it's that the cells are really liking that material and growing even more.
Interesting. Okay. Could you also use that to evaluate a surface, like different, let's say, different electrode materials? Although I guess in a cell or in an application you wouldn't actually do that, but
I think so. The actual material itself, we don't vary, like it's manufactured, right? Mm-hmm. But we do with the different coatings, right? So that's
right. Okay.
So in a sense, you can evaluate different coatings. Yeah. Of course, you don't want a coating that is too thick that will limit that detection of the impedance. Mm-hmm. But with our coatings, that hasn't been an issue.
Yeah, the layer by layer definitely seems like it's well suited for that.
Yep.
Awesome. I think this is the last question unless another one comes in. So on the slide where you had the 3 different donors,
okay.
Do you have any idea why donor 3 didn't show any improvement with collagen heparin?
No idea, and it's fascinating to me. So these cells, so these are commercial cells, so we buy them.
Oh, okay.
But we buy them, but they give us limited information about the different donors. So we have maybe the age and the sex, and that's about it. Gotcha. But in the literature, and they say that they're healthy donors, right?
Right.
But really, and I guess I failed to mention, these are human cells.
Okay.
But in the literature, there's a lot of literature out there that shows that really there's a lot that influences this type of behavior. Sure, age will influence, sex will influence, but also race is known to influence. Level of fitness has known to influence. Of course, if you have a disease, that's gonna influence the behavior of the cells. So yeah, it'd be great if they would provide more information, but at the same time, you need to keep some confidentiality. So that makes it a little bit more challenging.
Yeah.
But yeah, we don't know. But I find it really fascinating that they behave so differently. But
yeah.
2 out of 3, we call it a win.
Oh yeah, absolutely. So is it something that you could, like, let's say you see promising results like this?
Mm-hmm.
Could you is that something that would be examined in like a larger study, basically? Like,
Yeah,
if
I mean, I
if
would
you're
think
going to do
so.
something with it, you could, you develop uh, a product of some kind, and then you basically
Mm-hmm.
do the human trials, and in that case, you have all that additional information.
Yeah. Yeah. I mean, I would
Okay.
think so. And in fact, I mean, if we wanted to, and really, if we had the money,
Yeah.
we could, even with the cells that we have, we could do all sorts of like RNA sequencing or large, proteomic studies
Hmm.
to just sort of see that genetic signature of each of the different donors and see if we can correlate some differences in the genetic signature to the behavior that we're seeing.
That makes sense.
So that's something that I'm quite interested in doing. I just need the money to do it.
Sure. Yeah. Always. Okay, that's really cool. I think that's all the questions we've got.
All right.
So I will say thank you again for a fascinating talk. This is
Yeah, thank you.
really, really interesting stuff. Like it's very cool. I don't know, like that's really all I can say. It's just really neat. So I appreciate you sharing it with us. If I can get you to hang around for just 1 minute, we'll talk very briefly, and I'll let you go. So thank you to everybody for coming out tonight. We will be back again next week, same time, same Twitch channel. And we have Dr. Sumit Sharma from Ohio University who will be out here next week. So I hope you will all come back and join us again. And in the meantime, have a great weekend. And we'll see you then, I hope. Have a great evening, everybody.